We should begin.
Yeah.
Hello, everyone, and welcome to the Bioprotocol Roundtable webinar today. My name is Swapna Naik, and I'm currently working at the Yale University. My research focuses on understanding the role of RNA modifications in cancer homeostasis. At Bioprotocol, we are a group of early career researchers representing Bioprotocol as ambassadors of reproducibility and open science initiative. We truly believe that direct communication about scientific methods among scientists truly improves research reproducibility. In our working group of cell culture techniques, we are a group of 3 ambassadors. One of my colleagues, Jonathan, wasn't able to join us today. I would like to introduce you to Sherry. She is a graduate research assistant at Indiana University, and her research focuses on studying the cytokine signaling and airway allergic inflammation. So the objective of conducting Bioprotocol webinar is for sharing protocols, exchanging ideas from experts with diverse backgrounds, and discussing cutting-edge life science technologies. And we have a very interesting speaker on board today, Dr. Jilu Zhang. He's currently working as an assistant research professor at the Department of Microbiology and Immunology at the Indiana University School of Medicine. So Dr. Cheung received his Ph.D. from Jilu University, China, and later moved to the United States to pursue his postdoctoral research. He was initially at the Mount Sinai, followed by Indiana University, and his research studies have been very much focused on developing myeloid-derived suppressive cells-based cellular therapies for diseases as graft versus host diseases, that's GVHD, and graft versus leukemia, that's GVL, and exploring the potential underlying mechanisms. His research interests are therefore mainly focused on understanding and characterizing immune cell responses and developing innovative immunotherapeutic approaches for clinical conditions as GVHD and GVL, and also hematopoietic malignancies. He's also interested in screening CAR T cells for hematopoietic and non-hematopoietic malignancies. He, in addition, has received several honors and awards. He received the Spiritual Civilization Prize and Scholarship for Academic Achievement at the Jilu University, China, and also the American Association of Immunologists Young Investigator Award. And he has also published in several peer-reviewed articles. So in his talk today, he is going to walk us through general immune cell culture workflow and then focus on human T cells, culture for CAR T cell therapy in preclinical models. He's also going to talk about human mouse macrophages and myeloid cell culture and their downstream applications such as drug screening. I would also like to request all the attendees and participants, in case if there's something you did not follow or if you have any questions, please feel free. There is a chat box called Q&A, so please type in your request there. So now let's all please welcome Dr. Jilu Zhang and let him start his webinar. Dr. Zhang, please welcome to Bioprotocol Roundtable webinar today.
Thanks for the introduction. And today I'm going to talk about some primary immune cell culture and some downstream application based on my research. So before we talk about immune cell culture, I would like to give a little bit of background of immune cells. So there are 2 types of immune cells, innate immune cells and adaptive immune cells. The innate immune cells, including like monocyte, macrophage, dendritic cells, mast cells, or granulocyte, or NK cells. All those cells can initiate an innate immune response. Well, adaptive immune cells, including the B cells and the T cells, which can mediate adaptive immune response. So there are some cells can initiate both innate and adaptive immune response, such as NK T cells or the gamma delta T cells. So those immune cells can also divide into 2 groups, the myeloid cell group or lymphoid cell groups. So most of the innate immune cells are myeloid cells, except NK cells. And those myeloid cells are derived from myeloid progenitor cells. Well, all the adaptive immune cells, like B cell T cells or NK or gamma delta T cells, they are lymphocytes, which are derived from lymphoid progenitor cells from bone marrow. So to culture the immune cells, we summarize the workflow into 5 steps. The first step is to to have the primary tissue preparation. So we can get immune cells from such as the peripheral blood or cord blood, or even the bone marrow, or some lymphoid organs like spleen or lymph node. So after tissue preparation, we need to isolate cells from those tissues. So we can either use microbeads or use the flow sorting to get the immune cells we need. So after cell isolation, it's very important to check the purity of those immune cells. So we can either check their morphology under the microscope, or we can, I think the best way is to check them by flow cytometry, because those immune cells express their unique markers on their surface, such as CD3 for the T cells, CD19 for the B cells, and the CD14 for the monocyte, and the CD56 for the NK cells. So after the after validation of the purified T cell, purified immune cells, then we can culture them in the different media with the growth factors and the cytokines in the either flask or plate. If we need a large scale production of those immune cells, we can use a more advanced culture system that I can discuss a little bit later. And after we get enough cells, we can use those cells for the drug screening or for the gene editing, or we can also transfer those immune cells into the animals and study their in vivo functions. So in humans, the most common tissue source for the immune cells are peripheral blood, or we can also get the cells from blood leukocyte. So leukocyte actually is derived from peripheral blood. It's a byproduct from blood donation. It is also called Buffy code. It is enriched leukocyte. So in addition to the peripheral blood, we can also get the cells from cord blood or bone marrow cells. So, but we usually have limited source to the bone marrow or the cord blood cells. So for the culture of eosinophil, mast cells, and the basophils, so because those three types of cells are a very minor population in the peripheral blood, it is not practical to directly isolate those cells from bone marrow or from blood. I think and also those cells are terminal differentiated cells, so they won't proliferate in vitro. So the most common method to culture these cells is to isolate CD34 positive stem cells from bone marrow or from cord blood, or even from mobilized peripheral blood, and then differentiate those CD34 positive stem cells into those cell types with different cytokines like IL-5 or IL-13 or stem cell factors. So for dendritic cells, so you can directly isolate dendritic cells from peripheral blood. However, the yield it will be very low because DC is also a minor population in the peripheral blood. But for the macrophage, it's almost impossible to isolate the macrophage from peripheral blood because most of the macrophage are located in the tissue. So the common method for the DC and the macrophage culture is to differentiate the monocyte into those two populations, because it's relatively easy to isolate monocyte from peripheral blood. So that's why people usually isolate monocyte from peripheral blood and then differentiate them into the DC or macrophage. So the neutrophil is a different story because the neutrophil is the major population of the peripheral blood. We can directly isolate from the peripheral blood. So for the culture of the lymphocytes, such as T cell, B cells, or NK or gamma delta T cells, so because those cells can have the proliferation capacity in vitro. So after stimulation, those cells can proliferate. So we can directly isolate those cells from peripheral blood, then stimulate those cells, get larger amount of those cells. So those are the human common cells. So for the mouse, it's relatively for the mouse, we can culture those myeloid cells from bone marrow. So we harvest the bone marrow cells and then differentiate them and then differentiate those bone marrow cells into those myeloid cells in presence of different cytokines. And for the DC and the macrophage differentiation, we don't need to isolate monocyte. We just need direct differentiation from bone marrow cell. So for lymphocyte, we can directly isolate from lymphoid organ, such as spleen or lymph node. So in today's talk, I will not go through the protocol for all of those immune cells. So instead, I will mainly focus on human and mouse macrophage culture for testing some therapeutic antibody for the cancers, potential therapeutic antibody for the cancers. And in the second part, I was talking about some mouse myeloid-derived suppressor cells. We call it MDSC culture for the cellular therapy. And in the last part, I will talk about human T cell culture for the CAR T cell therapy in the pre-clinical models. So first, I'll start with human macrophage. As I mentioned in the background, so in order to culture macrophage, we need to isolate the monocyte. We start from human peripheral blood. So we isolate the peripheral blood mononuclear cells, we call it the PBMC, from peripheral blood. Then isolate monocyte from PBMC. So here is a detailed protocol, detailed procedure we did. So first, we dilute the peripheral blood with PBS, and then load those dilute blood on a separation media. So usually we use a cycle, and then we centrifuge the blood without break. And after centrifugation, we will have several layers in the tube. So on the top, we will have the plasma, and the middle layer has a platelet and PBMC. That's what we need. And then the bottom layer are enriched with red blood cells and some granulocytes. So when we get the PBMC, we can use microbeads to isolate monocyte. Here we use the CD14 microbeads because CD14 is the marker for the monocyte. Or you can also use other commercial available microbeads to isolate the monocyte. Here is an example show the purity of the monocyte. Here it shows before purification, the percentage of monocyte is only about 26%. After purification, you can get more than 90% purity. So I want to mention that that's not the only way to purify the monocyte. You can also purify the monocyte without beads. Just simply put the PBMC on a Petri dish and let the monocyte attach to the button, and then wash out the non-adherent cells. But that method is not very reproducible compared to the beads isolation method. So after purification of the monocyte, then we put the monocyte into the media with MCSF. So MCSF is a key growth factor for the macrophage. After 3 days culture, we will get a macrophage. We call it M0 macrophage. So the M0 macrophage can further differentiate into the M1 macrophage in presence of interferon gamma and LPS. Or we can get M2 macrophage when we culture the M10 macrophage in presence of L4. So here is a morphology. Here's a morphology of the M1 or M2 macrophage. Here I didn't show M0 because the morphology of M0 macrophage is quite similar to the M2 macrophage. As we can see in the human M1 macrophage, the cell looks like a round shape. Well, but in the M2 macrophage, you can see the cells are elongated. It's like a spindle shape. So there are significant difference between M1 and M2 human macrophages. So as we know, the M1 macrophage is known for its pro-inflammatory activity, and it also has enhanced antigen presentation compared to M2. And it also can mediate the tissue damage, and it also has antimicrobial and anti-tumor activity. Well, for the M2 macrophage, it's known for its anti-inflammatory activity and enhanced phagocytosis, and also it's involved in the tissue regeneration and angiogenesis. And it also can promote tumor formation and tumor progression. So now we have those tumor. Now we have those in vitro cultured macrophage. So we would like to use those macrophages to test an antibody that can be potentially used for the cancer treatment. So this antibody is an anti-LILRB2 antibody. So this antibody target on LILRB2. So LILRB2 is inhibitory leukocyte immunoglobulin-like receptor 2. So it is an inhibitory receptor widely expressed on the myeloid cells, including the macrophage. So how does this receptor regulate macrophage function? So as shown in this figure, so the LILRB2 interact with its ligand called angiopoietin-like proteins. So here I want to mention, so this is only one of ligand for the LILRB2. So the LILRB2 signal will transduce inhibitory signal through the atom motif, and which will induce phosphorylation of SHP1. And this phosphorylation will lead to a M2-like phenotype. So those M2-like phenotype, macrophage can produce like arginase 1, IL-10, or TGL-beta. So in our assay, we try to use an anti-LILRB2 antibody to block the RB2 signal, then prevent phosphorylation of the SHP1. Then will inhibit M2 phenotype, will favor a M1-like phenotype, such as enhanced TNF-alpha production or adenosine production. So based on those observations, we hypothesize that the LILRB2 antagonism will reprogram the tumor-associated macrophage and promote anti-tumor immunity. As we know, as I mentioned, the M1-like macrophage are more anti-tumor, and the M2 macrophage are more pro-tumor. So you may ask why we didn't use tumor-associated macrophage for this assay, why we need to use the in vitro culture macrophage. That's because we have hundreds of antibody. We won't get enough tumor-associated macrophage from patients for this screening purpose. So here's the assay we perform. So first we culture the macrophage, M0 macrophage, and then we add the antibody to the cell culture for 48 hours stimulation, and then check the phenotype of the macrophage. In another assays, we would like to see how does the antibody regulate M1 or M2 culture differentiation. So we have this M2, M0 macrophage, then culture those macrophage in the either M1 culture condition or M2 culture condition in presence of anti-LILRB antibody. So after 48 hours, so we first can check the morphology changes of the macrophage, and then we can harvest the cells and do some flow analysis to check M1 or M2 markers on the surface. And we can also re-stimulate those cells with LPS and harvest the supernatant to test the M1, M2 signature cytokines such as TNF-alpha or IL-10. And furthermore, we can also isolate mRNA from those treated or untreated cells and did RNA-seq to do the analysis on the transcriptomes. So in today's talk, I will not show all of those data. I will only show some representative result that has been published. So first, we'll start with the M0 macrophage. As we can see, as I mentioned before, the M0 macrophage looks similar to M2 macrophage. They have this spindle-shaped morphology. However, when you add an anti-LILRB antibody to the culture, you can see the cells tend to a round shaped, indicating there are some kind of changes in the cells. So we further check M2 marker CD163, which is a M2 marker. It's a scavenger receptor response for the phagocytosis. As we can see, the anti-LILRB2 antibody significantly decrease this M2 marker expression compared to the control group. So we further check the signature cytokines from the macrophage culture, TNF-alpha and IL-10. As we can see, the anti-LRB antibody significantly increase the TNF-alpha production will inhibit IL-10 production. So those data suggest that the anti-LRB antibody can promote M2-like phenotype in the macrophage. So next, we check whether the antibody can affect the M1, M2 cell differentiation. So as shown here, we checked another M2 marker called DC sign, which is involved in the immune tolerance mediated by the M2 macrophage. We can clearly see that anti-LRB2 antibody significantly inhibit DC sign expression in both M1 and M2 macrophage. So here is the statistic analysis. And we also check the PD-L1 expression because as you may know that PD-1 are involved in the immune suppression. So as we can see, the anti-LRB2 antibody also significantly inhibit PD-L1 expression. Because in M2 culture, in vitro culture M2 macrophage, the PD-L1 expression is very low. We couldn't see a significant difference in the M2 culture conditions. So we also did some RNA-seq, which I will not show today. It also show that anti-LRB antibody can promote at least M2-like phenotypes in the macrophage. So here are the human macrophage. Now, how about mouse macrophage? As I mentioned in the background, we can get some macrophage from bone marrow cells. So we got some bone marrow cells from mouse femur, and then culture those bone marrow macrophage in presence of MCSF. The protocol are quite similar to human macrophage. After 3 days culture, we got this M0 macrophage. Then we can further differentiate those M0 macrophage into M1 macrophage with interferon gamma and LPS, or M2 macrophage with IL4. So here's a morphology of mouse M1 and mouse M2 macrophage. So different from human macrophage, it's a little bit different to distinguish between M1 or M2 from the morphology changes. However, we can still harvest the cells and check the marker expression on the M1 and M2 mouse macrophage. So it's also important to check those markers because when we differentiate macrophage from bone marrow, and the bone marrow is a mixed population, it may have some other cell contamination from the bone marrow cells. So we track the several surface marker on both M1 and M2 cells, such as the CD11B. As we can see, both M1 and M2 mouse macrophage express CD11B, and the CD11B is a myeloid cell marker. We also check the key marker for the macrophage F4-80. Again, we can see both of them are highly expressed F4-80. We can also see that the most of cells are macrophages. We can also check some other markers like M2 marker CD206, which is a mannose receptor. Is highly expressed on the M2 macrophage. As you can see, the M2 macrophage express higher CD206 compared to the M1 macrophage. We can also check some antigen presentation related molecules such as CD86 or MHC class 2. You can see that M1 macrophage express higher CD86 or MHC class 2 compared to the M1 macrophage. We can also check the PD-L1, different from the human cells. Both M1 and M2 mouse macrophage highly express the PD-L1. So we also check some monocyte markers, so like a CD115. As you can see, actually, once the cell differentiate into the macrophage, so they are downregulated. They are downregulate the CD115. So we also check the LRB2 expression on the macrophage. As you can see, they are highly expressed. So it should be noticed that LRB2 is a human receptors. The reason the macrophage can express those human receptors because we isolate the bone marrow cells from transgenic mice. and those transgenic MACs are highly express LRB2. So we would like to use those cells for screening of our anti-LRB2 antibody in the mouse system. So there are some ongoing experiment for this testing, so which I will now show it today. So in the second part of my talk, in the second part of my presentation, I will talk about mouse myeloid-derived suppressor cells culture for the cellular therapies. So we call. So I didn't introduce the MDSCs because MDSC is not a normal immune cells. So MDSC is almost undetectable in healthy individuals. MDSC is a heterogeneous group of immune cells from the myeloid lineage. And MDSC can only be detected in the pathological conditions such as chronic infections or cancers. So MDSC, as its name, it's a suppressor cells. It can interact with other immune cells such as T cells and inhibit their function, such as T cell activation or proliferation, or their cytotoxicity. So there are two types of MDSC based on their morphology. So monocytic MDSC or granulocytic MDSC. So there are certain markers for those two types of MDSC, such as LY6C or LY6G. So there are many protocols to generate in vitro cultured MDSC. So in our study, we generate those MDSC from bone marrow cells. So we got those cells from bone marrow cells from the mouse femur, and culture those cells in presence of GM-CSF and IL-6 for 4 to 5 days. So there are some other protocol also using the GCSF for generation of granulocytic MDSC. But in our study, so those are sufficient to generate those suppressor cells. And this protocol is also well published in minor high impact journals. So after, as you can see, this is the morphology of the MDSC. It's a heterogeneous population. They have some adherent cells, and they also have some cell looks like also have some suspension cells, and some are elongated shape, some are round shapes. So since we want to use those in vitro cultured MDSC for cellular therapy, the first thing we need to do is to check their function to confirm their suppressive activity. So we did some in vitro cultures to test their functions. The first we cultured the bone marrow derived MDSC with CFSE labeled T cells. The CFSE, if you are not familiar with CFSE, I will introduce in the next slide. So, and then we stimulate those cell culture with anti-CD3 or anti-CD28 antibody. So those two antibodies will induce activation of T cell activation and the proliferation of T cells. And after 3 days culture, so we will check the proliferation of the T cells by flow cytometry. So in this assay, we culture the T cells and MDSCs in different ratios from A to 0 to A to 2. So after 3 days culture, when we check the size of the colony, we can clearly see a difference. So you can see the colony from the group with MDSC are significantly smaller than the group without MDSC. So if we add more MDSC, so the size of colony is even smaller. So then we harvest the cells and check the proliferation by flow cytometry. So in case you are not familiar with the CFSE, the CFSE is a fluorescence dye. It can label any cells, including the T cells. So once the T cells was labeled, so it will have fluorescence can be detected by flow cytometry. When T cells is proliferating, so the fluorescence of CFSE, the fluorescence intensity of CFSE will decrease. And when we check by flow cytometry, we will see a different peaks of CFSE staining. And those those peaks represent the cell proliferation. More peaks means more proliferation. So in our study, since we are using total T cells, so we can check both CD4 and the CD8 T cells in our cultures. As you can see here, so in this plot, I didn't show the histogram because I also want to see the CD4, CD8 staining on the cells. So we can see without MDSC, after three days culture, about 74% CD4 T cells are proliferated cells. About 90% of CD8 are proliferated cells. However, when you add MDSC in the culture, the proliferated cells are decreased to 50% in the CD4 and 70% in the CD8. So if you add more MDSC in the culture, the percentage of CD4 and CD8 T cell are also significant decreased. So those data suggest that in vitro cultured MDSC can inhibit T cell proliferation in vitro. So next, we want to test their in vivo functions. So we use a graft versus host disease model, or we call it GVHD. So just a brief introduction for the GVHD. So GVHD is the major complication after allogenic bone marrow transplantation, and alloreactive donor T cell activation are crucial for the tissue damage in the GVHD, and the inhibition of T cell activation will alleviate GVHD. So given those background, we want to we hypothesize that MDSC based cellular therapy will ameliorate GVHD. Here is our protocol. We first irradiate the mice. It's a lethal irradiation to get rid of all the immune cells, and then we adaptively transfer allogenic T cells and the bone marrow cells to the mice. So it's IV injection. And at day 0, day 4, and day 10, we will adopt transfer MDSC. So we will give 3 doses of MDSC cellular therapy. So as you can see, in this study, we also have a control group. So to make sure that the mice would not die of irradiation, in this group, we only transfer the bone marrow, and the mice from this group will not develop GVHD. But for the other group, we will transfer both allogenic T cells and bone marrow. As you can see, the mice without MDSC transfer only have about 20% survival rate. Well, the mice receive MDS transfer has about 60%, suggesting that MDSC prolong the mice survival in this GVHD model. So this is MDSC mismatch model, and we also test the MDSC in a MHC match model. We see a similar result that MDSC can significantly prolong the mice survival in the GVHD model. So all of these results suggest that in vitro cultured MDSC that can be used as a cellular therapy to treat graft versus host disease. So in the last of my presentation today, I will talk about human T cells culture for the CAR T cell therapy in preclinical model. So in this part, actually, unpublished study. So a little bit background for the CAR T cells. So the CAR T cells the CAR T cells are chimeric antigen receptor T cells. So in order to get CAR T cells, so we will need to either use a retrovirus or electroporation to introduce the gene of CAR into the T cells. So after transduction of the CAR construct, the CAR T cells will express the CAR construct on the surface. So in the CAR construct, there are mainly 2 components: the extracellular domain, which is antigen recognition domain, it is derived from it is a single chain variable fragment derived from antibody. So it also has an intracellular domain, which is a signal transduction domain derived from CD28 or 41BB. Those two molecules are well known for its cause. Those two molecules are the co-stimulation molecules in the T cells. So how does T cells kill tumor cells? So the T cells can recognize the tumor antigen on the tumor on the surface of the tumor, and then attach to tumor and form immune synapse. And this immune synapse will lead to make this CAR T cells perform cytotoxicity against the tumor cells, so result in death of the tumor cells. So because our CAR T cell, because our CAR T study is unpublished, in today's talk I will not release what kind of tumor antigen we are targeting, but I will discuss the protocol, how we generate those CAR T cells, and how we validate those CAR T cells function in vitro and in vivo. So we start from the blood leukocyte from healthy donor. So this can either get from a blood bank, or you can order from commercial shops. So we isolate PBMC from the peripheral blood, or we can also isolate the T cells. We actually did a comparison between the PBMC versus purified T cells. They are quite similar. Actually, we in our study we only use a total PBMC cells. So during the purification of the cells, we need to coat a plate with anti-CD3 and the CD28, which I already mentioned that it's used for activation of the T cells. So and then we add this PBMC into this coated plate in presence of IL-7 and IL-15. So there are some other protocols for the CAR T cell culture. So they are using a high dose of IL-2. But in our protocol, we are using this IL-7, IL-17. It's already shown that those protocol will induce less exhaustion of the T cells. So after 48 hours, we will generate those activated T cells. So during this 48-hour waiting period, so we need to prepare a retronectin coated and virus loaded plate. So we first prepare this retronectin coated plate. Retronectin is actually a fibronectin fragment. It will enhance the transduction efficacy. So we just coat the retronectin to a non-tissue culture treated plate, and then we add lentiviral supernatant to the plate, and then centrifuge and let the virus attach to the plate, and then we remove the supernatant. So we generate those lentivirus from amphophenic cells, which is engineered 293 T cells, and these lentivirus are pre-packed with the genes for the CAR and the GLP. We use GLP to check the cells in vitro and in vivo. So after we prepare these pre-coated cells, we add these activated cells into the plate, and after 24 hours, so we will generate these CAR T cells. So then we can further extend those CAR T cells in non-tissue culture treated plate in presence of IL-7 and IL-15. So if you need a large scale production of CAR T cells, you can use some more advanced culture system. So here we use this G-Rack system. So in this system, in this cell culture containers, there is a membrane in the bottom of this container will allow cells has a better air exchange. So then we can we can produce a large amount of CAR T cells. So after amplification of CAR T cells, we can freeze those cells in the liquid nitrogen, and then sell them for any in vitro study or adopt transferred mice for the in vivo studies. So before we use these CAR T cells, the first thing we need to do is to check the transduction efficacy of those CAR T cells. Here is an example. So in this experiment, we have untransduced T cells. We call it control T cells. We also have CAR T cells with transductions. As you can see, because we are using the total PBMC, actually we can also say the total T cells. We have both CD8 and the CD4 T cells. As you can see, the control group in the cells in the control group has no GLP expression, but in the CAR T cell group, they have about 80 to about 80% GLP expression, indicating there are about 80% T cells are CAR T cells in this culture. So we further validate so after validate the CAR expression on the CAR T cells, we now need to check their functions. We want to know whether they can kill the tumor cells. So we in vitro culture the CAR T cells with leukemia cell lines, which is MV411. So this tumor cell line expresses the tumor antigen that can be recognized by the CAR T cells. So this is a short-term cytotoxicity assay. We just culture the CAR T cells with we culture the T cells with the tumor cells in different ratio from 10:1 to 1:1. As you can see in the 10:1 at 10:1 ratio, the CAR T cells has about 80% cytotoxicity against the tumor cells, while the control T cells, do not have too much cytotoxicity. You may question about the cytotoxicity are not that strong because you need 10 CAR T cell and versus 1 tumor cells. That's because this is only a 7-hour assay. We also have a long period culture assay for the in vitro assay. We culture the CAR T cells with tumor cells, and then we found even in the 1:1 ratio, so the CAR T cells can completely eliminate the tumor cells in 48 hours. So those data are just in vitro assay to confirm the CAR T cell function and to confirm the expression of CAR on the T cells. So the next we want to validate their in vitro functions. So we use this immunodeficient mice, which the strain is NSG SGM3. In these mice, they do not have any they don't have any T cell, B cells, or NK cells. So but they do can produce human cytokines such as ICF, GM-CSF, and IL-3. So those cytokines will support the human cells that transfer into the mice. So this model is also widely used for the CAR T studies. So here is the protocol. At day -5, we transfer 1 million leukemia cells, MV411, and this MV411 leukemia cell also has a luciferase expression that allows us to check the tumor burden. So at day 0, so we did bioluminescence imaging. So using this IVS imaging system, as you can see, those bioluminescence in the mice, actually those bioluminescence represent the tumor burden, and this is before the CAR T cell transfer. And it's interesting to see that actually, although this is leukemia cells, it seems the tumor, the leukemia cells accumulated in these two areas. We don't have an answer for that, why it shows this pattern, but we can clearly see that all of mice has a similar tumor burden. So after confirmation of the tumor burden, so we adopt transfer 5 million CAR T cells or control T cells. We also have some mice receive just a PBS. At day 0 after this imaging, as you can see, this is the grouping. We have 2 mice PBS control group and 3 mice in a control T cell group and 5 mice in the CAR T cell treatment group. So after cell transfer, we perform the weekly bioluminescence imaging and to check how that T cells kill the tumor cells in vivo. So here is the result. As you can see, at day 7, the tumors start to grow. In the PBS group or control T cell group, and you can already see the CAR T cells start to eliminate tumor cells. As you can see, the tumor burden seems to be lower than at day 0. At day 14, and the tumor cells are continuous growing in the control group, but in CAR T cells, you won't see too much tumor cells. Well, at day 21 or day 28, you can see a massive tumor growth in the control group, but no tumor growth in the CAR T cell treated groups. So we actually follow the mice until 60 days. So we still are able to detect any tumor signal from the mice. So the statistical analysis show that actually the CAR T cells actually can significantly reduce the tumor burden. So all of those results indicate that CAR T cell, the human CAR T cells we generated can eliminate, establish the leukemia in vivo. So in summary, so the primary immune cell culture is a great tool to generate immune cells for the adaptive cell therapy, or for the drug discovery or cytotoxicity, like we discussed today. So the primary immune cell culture is also very useful to study the biology of the immune cells, and also the immune regulation in a different disease. So in addition, the primary immune cell culture can also use for generation of the monoclonal antibody production. In our study, the anti-LILRB2 antibody was generated by a primary culture of the B cells with hybridoma cells. Furthermore, actually, immune cell culture can also be useful for the vaccine manufacture, such as the DC vaccine. So we can in vitro culture the DC and load with tumor antigen and transfer them to the tumor bearing individuals. And also the immune cell culture can also use for biobanking to preserve or keep some pressures or some unique TCR or BCR clones of T cells and B cells. So we believe the future direction of the immune cells is the 3D cultures. So in the 3D cell culture system, including those scaffold-free system like organoid, also some scaffold-based system like bioprinting materials, and also this hybrid culture system like organ on a chip. So this 3D culture system will help us mimic the in vivo condition and allow us to study the immune cell interaction, to study the interaction between the immune cells and other non-immune cells. And I think that will be let us better understand the biology of the immune cells and the immune response in the physiological or pathological conditions. So at the end, I would like to thank my mentor and my previous mentor, Dr. Shusha Chen and Dr. Ping Pan at Houston Methodist Research Institute. Actually, most of the work I present was done in my previous lab. Because I just recently moved to the Indiana University, I also would like to thank my current mentor, Dr. Mark Kaplan, and the founding agency. So I will stop here, and I'm happy to take any questions.
Well, thank you, Dr. Zhang, and that was a very interesting talk and very fascinating work. So I have a couple of quick questions. So when it comes to using primary cells in adaptive cell therapy, so how important it is to determine the cell morphology in terms of all these cells, and what are the challenges that you usually face with?
Well, actually, so it's not that easy to determine the cell quality by morphology. So because, for example, the T cells, because they are suspension cells, it's hard to see their morphology changes. But I would suggest using as I suggest in the presentation, I would suggest to using flow cytometry to determine the quality of the cell culture. Just use the specific immune cell markers to check the cell quality.
Okay. And you also mentioned in the CAR T cell therapy using the IV injections, and there was a specific cell number you talked about in the initial time of 1 million cells, and then after 30 days, I think it was 5 million cells or something. So what factors decide these particular cell numbers when you do these IV injections?
Well, well, that's a good question. So the cell number for the tumor cells and the CAR T cells were determined based on previous published studies, because there are lots of study on the CAR T cells. But we also did some pilot experiment to determine what's the minimal cell number we can use to eliminate established tumor models.
Right. Sure. Thank you very much. And I would like to ask Sherry to take over the Q&A from here.
Yeah, so in the chat, we've got several questions for Dr. Zhang. Yeah, thank you so much for giving us like a really awesome talk. And we've got a question from Renee asking like the generation of human CAR T cells is fascinating. So what approximately is the efficiency of the lentiviral transfection of the PBMC?
Well, actually, we are not transducing the PBMC, we're transducing the T cells. As I shown, so in our method, so we're using the retronectin. I think that's the most common used method to do the transduction in the hematopoietic cells. The T cell is the hematopoietic cells. So as I shown in our studies, it's about 70 to 80% of transduction efficacy, but sometimes it may be lower, maybe 50%. But it really depends on the condition you are using. And also the media I didn't mention. So the media we use for the cell culture is, well, some studies show that you can just use regular RPMI media for the CAR T cell culture. But in our study, we are using the ex vivo media 15, which is a serum-free media. I think that may be help for the generation of the CAR T cells.
Yep. So the same question, like coming from Renee, is asking, like, can you use smaller antibodies such as single chains antibody fragments or fragment antibodies for some of your works?
Can you repeat the question again?
So can you use smaller antibodies such as single chains antibody fragments, the scFVS or FABS for some of your work?
Well, I think the golden standard of the CAR T, I mean the antigen recognition domain is from a single chain variable fragment. So it is derived from an antibody, but it's only a fragment. It's not a single chain antibody or a small antibody that you mentioned. I think that's a golden standard using ICFV other than the other component from antibody.
Yeah, so I've asked a naive question on the chat. Like, so everyone is welcome to ask, like, it doesn't matter like what kind of questions you ask, like even naive question is like welcome here. So why do we have to culture CAR T cells on non-tissue culture plates instead of like using tissue culture plates?
Yeah, that's a good question. So we know that T cells is non-adherent cells. They are suspension cells. However, after long-time transduction, some cells actually become adherent cells. Actually, when we harvest the cells, we need to either pipetting or use that or need to scratch the cells. The reason we use non-tissue culture treated plates is because we want to avoid adhesion of CAR T cell to the plate. It will help us to get more T cells from the plate.
Yeah, then I've learned something new now. Yeah, so we've got another question from Adu asking, are your CAR T cells CD8 T cells only, or they are both CD8 and CD4 T cells?
So they are both CD4 and CD8, as I already shown in this here. Because we are start from PBMC, I think most CAR T study are from total T cells, not from either CD8 or CD4. Because if you only use CD8, so that after you transfer those CD8 T cells, they may quickly become exhausted because CD8 T cell also needs some cytokine secreted from CD4. So that's why I think all the CAR T study using total T cells has both CD8 and CD4 T cell.
Yeah, thank you, Dr. Zhang. So do we have any questions other than chat? Like, you're welcome to like talk as well if you have any more questions. So if we don't have any more questions, maybe we have to come like as a closing from Swapna.
Yes, definitely. So thank you so much, Dr. Zhang. That was really a wonderful presentation and so much of innovative techniques and a lot of information. I got to know about the different aspects you're using in your research. It's not just diverse and versatile. And I'm sure everyone had a really good overview of your work and got to know about so many different things. So anybody has any further questions, I'm sure, are you okay people contacting you later by email or something? Are you open to that?
Feel free to contact me if you have any questions or if you want to have some troubleshooting for the immune cell culture.
Yes. And I would like to thank everyone for participating and all the organizers of the technical team from Bioprotocol especially. And I would really like to thank my team. And I would like to thank again Dr. Cheung for your precious time and coming in for the webinar today. Thank you so much.
Well, thank you. Thank you too.
Thank you.
Thank you.
You enjoy the rest of your day. Thank you.
You too. Bye-bye.
Thank you. Bye. And I would like to end the webinar. Thank you.